I'm very excited to introduce the next speaker, Professor Yanchao. Professor B is a principal investigator in the IDG McGovern Institute at Beijing Normal University, and she's a Changjiang professor in the State Key Laboratory of Cognitive Neuroscience and Learning. She got her PhD in the Department of Psychology at Harvard with Alfonso Karamazza, and her work focuses on the functional and neural architecture associated with concepts. To this end, she uses cognitive, neuropsychological, and neuroimaging techniques, including early on a fascinating case study of cognitive mechanisms linked to writing of Chinese characters. Today, she's going to overview the Beijing Normal University, and the title of her talk is The Science of Learning at Beijing Normal University.
Thank you. Thank you for the introduction. Today I'm speaking on behalf of my director, Professor Wu Li, who can't be here because of visa problems. So if you are director of the McGovern Institute, you are more likely, I think, a higher chance of having visa problems than the average. But I do appreciate the opportunity to talk to you, to introduce to you our BNU McGovern community. And it's a challenge to see how well I know my institute in 2 days. All right. So where did it start? It started 2010 when Pat, Laura, and Hugo took their first visit to our State Key Laboratory of Cognitive Neuroscience and Learning at BNU. And they liked us enough to come back 2 days later to meet with the president, the state council, vice premier, I think, and then got things rolling. A year later, the institute was formally established, and we have been carried through the board meeting every year since. And this is the Beijing Normal and our own institute. So this is our local community, the co-PIs and the lab members saying hi to you from Beijing, or a few of them here sitting in the audience, and our research. I think it resonates extremely well with the two McGovern directors here at MIT that we cover a wide range of research methods, computation approaches, electrophysiology, neuroimaging, and different brain and cognitive levels, looking at different higher order cognition and more basic perception problems, and especially with a focus on learning and development. So I was very happy that I'm speaking after Rebecca, and looking at different kind of human models and animal models. So BNU has a very strong tradition of education. So I'm going to share with you one of the central themes of the work we do is to understand learning and how to help learning. When we talk about learning, we really talk about many, many different things. So by learning, we can increase the perceptual ability, being able to see well or hear well. We can learn new associations between two different things or events. We can learn about episodic issues like what happened and when. And we can learn about different kinds of knowledge, like learning a new language, learning music, learning sports, learning math, or semantic knowledge about the whole world. And we also learn about social rules. Like this is the empress in this new clothes. So we can see well, we can understand well, but somehow the social rule tells us not to speak about it. So all these things require learning. And we have done every aspect of it, using mouse models, monkey models, and non-invasive neuroimaging in different groups and individuals. So I'm going to present one or two studies across the slices, and I hope it's not too much, but to give you a sense of flavor of what's going to happen. All right, I'll start with the most basic aspect. I'm starting with perception because poor guy, again, Wu Li, this is his work. He can't come, so I'm ordering him first. So perception is very important for survival. If you're not good, you can't see a leopard here. Can you? So that's very bad for us, like primates. And but apparently through training, you can see it. So with training, people are better at perception, like detecting the contours, seeing things that's like local contrast. The key question is what's happening? Like how does it work? What's the neural basis for perceptual learning? And a long-going debate is whether it's increasing the first-pass neural sensitivity of the vision or a later readout efficiency. So Liu's lab did this by recording from the primary visual cortex, V1, using multi-array electrodes when the monkey learned these tasks. So through learning, the monkey has to has to say whether there's a straight line. Okay, through learning, they can do these tasks. What they found? So they found basically something is happening in B1 already, the first pass of the cortex, when the visual signal comes in. So this is across days, the neuronal population decoding accuracy of the V1 neurons. You can see through training, it's better, it's getting better. Just looking at the neuron firing pattern, you can decode whether there's a straight line there. And such decoding accuracy correlates with behavior well. So it seems that V1 neuron is already shaped by this training. But interestingly, if you unpack through time, if you look at the time course, the first component, about 40 milliseconds, is not affected by training. It's a later response, about another 40 or 60 milliseconds later, the training induced a difference. So it seems that maybe the first response cannot be changed, but the readout process. They also did it on local contrast. Like, you're supposed to see which line is different. So after training, you become very, very good. And again, here, they replicate the previous findings such that the early component is not affected, but the later one. However, being in science, there's always this extra complication. So when they break down the trials by good trials and bad trials, they found that even the early component seems to be changed. So in the good trials, the neural firing, even for the early component, becomes stronger, it's more efficient at guiding your behavior. So they're still debating about this interplay, complicated interplay between bottom-up and top-down processes. Okay, we learn about perception. What about learning to remember what you perceived? So this is Xue work. They ask people very simple tasks, like to look at a bunch of faces, look at a bunch of words. You're supposed to remember who you have seen or what words you have learned. What they found was that a very good predictor of which face or words being remembered later on is how similar the neural activity patterns are across the repetitions. So suppose I see Laura a few times. So each time, if my neural response patterns are very similar, I'm going to remember her well. If during these several times it's not very similar, I'm going to forget her. So the neural similarity pattern predicts the remembering process. In their later work, they keep seeing this using different kind of techniques. So this one is combining stimulation technique and EEG. They found that not only the spatial pattern of your neural response is important, but also the temporal pattern is when they're firing together. And furthermore, if you stimulate your lateral prefrontal cortex, you can enhance that spatial temporal neural pattern similarity, and then leads to better remembering. So by stimulating the prefrontal, if you want to learn something, that's probably a good strategy. In this recent work they just published, is that this neural pattern similarity not only plays a role during your learning phase, but the similarity between the learning, the encoding stage, and the retrieval stage also plays a very important role. So again, they ask people to learn a bunch of words, and in the condition where there's not very good similarity in the visual cortex, during the learning part and the retrieval part, it leads to false memory. It's like, maybe I have learned this word, but you actually didn't. It happens when, for the visual cortex, the learning and the retrieval part, the neural pattern does not synchronize. Okay. So the first two parts, the perceptual learning, episodic learning, the similarity is the neural response pattern being important. We also made progress in identifying the structural basis of learning. So this is Xiaohui Zhang's work that they look at neural circuits. So they use optogenetics and neurophysiology techniques on mouse models. And firstly, they showed that they discovered a direct pathway from lateral entorhinal cortex selectively transfers synaptic information to hippocampus C1 that was not found before. And this direct circuit is necessary for learning the association. They call olfactory associative learning. So if in this mouse, this direct circuit is knocked out, these mouse are not going to be able to learn to associate one odor with a water reward. They're not going to be learn this new association. Okay. So very different things like learning about association, episodic perception in mouse and monkeys and humans. But we keep seeing some similar conversions. Like there's interplay between bottom up and top down, or encoding and retrieval is happening, makes a big difference. And the neural pattern similarity seems to be a very key role in predicting behavior, how well you learn. And we also keep discovering these new structural circuits. As opposed to the relationship between this direct link and the functional pattern similarity, we don't know yet. And these are the things that we're talking about in the lab. Okay. So when we talk about learning outside of this room, outside of the psychology community, people talk about learning this, like learning a new language, learning new knowledge, learning math. And we have done a lot of work on this too. I'll start with language, learning to read Chinese. So learning to read Chinese is hard, even for Chinese people. I have a kid who's starting school. I was like, this is hard. How did I learn? So what's the cognitive basis to support this learning to read development? So in this study, Professor Hua Shu, they followed 294 children, starting from age 4 for 8 years. And every year they test them on many things. What they discovered was that, I think Bob mentioned this too, the ability before you learn to read, in this case, the syllabic awareness is how well you're aware of the syllabic structure when you hear Chinese, is a very good predictor of how well you are aware of the morphological structure in a Chinese sentence after you learn to read. Okay, so the coupling the auditory process before you learn to read seems to be very important. And if you have a brain abnormality, in this case, if these two particular pathways are not as well developed, so they compared children with dyslexia in China and controls, they found that these people have less integrity in these two pathways, and the integrity of these pathways associated with phonological processing and morphological processing, respectively. And actually, another work in collaboration between Lu Chunming in our lab and Gabrielli at MIT, they were providing some conversion evidence for both dyslexia and autism, having problems with this particular inferior longitudinal facilicus. Okay, so it's hard to read, and we have reading dyslexia. How do we fix this? Bob mentioned this already, so I'm going to a little bit more detail. So this is a joint work by Bob Desmond's lab and Nanyun in our institute. So they compared training with music, learning piano, and learning to read in groups, and also control groups, and then look at how it affects children's ability to process auditory words. What they found was that with the music training group, it's even better than the learning to read group, enhancing the ability to do auditory discriminating tasks. And furthermore, look, the piano group has a stronger neural response to auditory cues, and that neural response correlates with behavior. I'm not sure I told it. Did I tell this right? Well, okay. You're not having a judging face. It's always challenging to talk about other people's work. Okay, so learning piano is good for your word processing. So that's auditory and motor, right? Learning visual perception is also good. So in this study, it's very cool. So they asked dyslexic children just to do the basic of visual perceptual learning, just to look at patterns and detect them. And it turned out these children improved on reading fluency. So they did not train on anything linguistic. They didn't train even on auditory. So it seems that reading really, using Bob's word, heterogeneous, like a complex system that intersects with all these basic sensory processing systems. Okay, so when we learn to read, we not only try to learn the sound of the words, but we also learn the meaning of the words. The meaning of words is really the knowledge we have about the whole world. Okay, look at the meaning of these words. Cup, rainbow, and freedom. Do they differ in anything? I'll tell you. So we were also curious about how important is sensory processing in learning about the meaning of the word or the knowledge about the world. So we contrasted congenitally blind subjects and controls to see how important vision is. Many people work on vision, and we know it's very important. But it turned out it's not that important for some things. Like in this case, we will ask people to judge about the shape of the cup. And you think, okay, the blind people, of course, know the shape because they can touch, right? So yes, it turned out they know the shape very well, similar to sighted, but not obviously in the brain, both blind and sighted has this information stored in the so-called visual cortex, in the inferior temporal lobe. So we talk about visual cortex, but it doesn't matter whether I'm acquiring this information through eyes or through touch, but as long as it's about the same kind of knowledge. Okay, but it's a different thing for rainbow. Because if we can't see a rainbow, we can touch it, we can smell it, we can play with it, right? How do we acquire it? It turned out for this part of the brain where more abstract knowledge is stored, rainbow for blind is like freedom for sighted. It becomes more abstract. Wow, we can still know everything about it. It's still stored there. So we think that it's learned based on language, based on verbal relations. Through language, we learn everything. Well, the way that the brain stores it becomes in the abstract part of the brain. So it's sensory important for learning language. Yes and no. It's not necessary to learn all these skills. Well, it affects where and how the information is stored. Okay, I hope I explained that well because that's my own work. Like everything else, we were curious about the structural basis of all these knowledge representation. So looking at the higher cognition is hard. So we took advantage of the lesion model in the brain, in the humans. So stroke patients with tumor, resection, TBI, and we identified 3 major white matter pathways, very long-ranged, being necessary for these. So if you have a lesion here, if your lesion is big, that will lead to semantic deficit. And the size of the lesion correlates with semantic deficit severity. Furthermore, using machine learning, representation similarity analysis, loss of decoding approach, we found that the pattern of your lesion in these white matter tracks can predict or decode the pattern of your semantic deficits, suggesting that all this fine knowledge might be stored in these long-range connectivity pathway. I'm still seeing a judging face from Bob. Okay, learning about language, reading, semantic knowledge. So what's the common thing? Again, we see tight interaction with sensory processes, higher cognition and sensory processing. We see that reading, so the meaning, the sound, and the morphological processing are both supported by different kinds of long-range connectivity patterns. Okay. Finally, learning about social. I was very inspired by Rebecca's talk about social because it's going to help me to explain. So this is Ina Ma's work, a very brilliant young PI, Imana. So she was always curious about social learning, social cognition. And I'm only presenting 2. She has many good work, only have time for 2. So one is learning about social information. So they ask people to do a very simple estimate of something bad happening to you. Then they give you about information in the social group, the probability of what's going to happen in the social context. And then they ask people how well they learn this information to modify your own behavior. It turned out people have a very strong positive bias. If the feedback is given in a positive way, they learn it very fast. They immediately change their own behavior. But if there is something bad, they don't care. It's like they're plane dropped, but it's not going to happen, the conversation we had yesterday. So people are very biased towards positive social information to change their own behavior. Furthermore, if you give them oxytocin, apparently a very classical social modulator, through nasal spray, it changes their behavior. But again, only on the positive information. They become even better at taking in the positive social feedback. Okay. And what about making complex social decisions? So this is a very simple task, but it's so common. So they ask people to judge, if you just simply give people money, you give some money to yourself and others, and whether this is a good way or bad way. And it's a social decision or judgment that varies by people. And it goes back to Bob's earlier point of precision. So what they found was that whether you judge this to be a good allocation strategy, it totally depends on your own internal reference point. So they did something very smart. They asked people to give the best strategy you think, and then lay this on this cognitive space, what they found was there's a huge individual difference. Some people are more selfish. They cares more about fairness. Some people are totally self-centered. I just want me to get more money. And when the new information comes in, is there that comparison of the stimuli, and that there's very different internal reference point. That angle, that error signal determines their behavior, whether they think this is a good plan or bad plan. So this huge individual variance would need to be taken into consideration when we see a behavior. And furthermore, that error signal, that cosine bar, is encoded in amygdala activity. So if that beta is strong, the amygdala activity is strong. And furthermore, again, the check is, if you give them oxytocin, it enhances the neuroamygdala representation. It then changes your behavior to more pro-social. You become less self-centered. You are more towards fairness. I think everyone should just take oxytocin. Or make decisions when you have higher, like during pregnancy. Okay, the final part of learning in social context. Again, it goes back to Rebecca's point. Learning by yourself in the social context by a teacher is totally different. So in this study, Lu Chunming, they scanned using F-nurse, two people, the student and the teacher simultaneously, to see what's the relationship between these two people's brain that's related to learning in the social context. Okay, long story short, again, neural pattern similarity between the teacher and the student is very important. But it's amazing, there's a time lag. So how well the teacher's neural pattern predicts the student's neural pattern, about 10 to 30, I forgot, milliseconds. If they can predict well, then the learning becomes more successful. It took me a long time to understand why. Because the prediction is really based on high-level internal structures. It's like the teacher has to be very sensitive of the cognitive state of the student and to modify it accordingly, modify how you teach accordingly. Okay, I like that sound. Okay, so I'm trying very hard to get some coherent message out of all these. So this one I said, okay, positive bias, error signal encoded amygdala affected by oxytocin, and the neural pattern similarity across individuals affects knowledge transfer, going back to the early kinds of learning study. All right. So what's the convergence? What's the divergence? So many different kinds of learning, different aspects, different levels. But I hope you're with me that you see something pops out. Like neural pattern similarity is important for perception, episodic, semantic knowledge, learning in context. And structural basis, many are related with long-range connectivity patterns. The big question is, what's the main general mechanisms of all these kinds of learning? Can we use some of the mechanisms to enhance learning, of helping people to learn better with language, to help people with learning difficulties? So these are the heated discussion we have in the lab. We're hoping to test some of the findings with mouse on like learning knowledge or the effects of oxytocin learning different kinds of things. And all these findings is part of the progress report to the support of the McGovern. So where are we going from this? We're going longitudinal. Going longitudinal is like a no-brainer if you're interested in learning. Well, I have to put more science in. We're guided by some basic science findings we had in the lab. Don't worry, it's going to finish very soon. So again, the mouse model, we keep getting more and more understanding about the importance or the mechanisms of critical period in development. For instance, it's still Xiaohui Zhang's work. They show that, okay, this inhibitory GABA regulates the binocular synchronization of neurons when you have a visual input. Just bear me with this. But it only happens during the critical period. It doesn't happen before or happen after. So something about the sensitivity during that time. And also inhibitory interneurons are involved in regulating the onset of that critical period. So inhibition system seems to matter very much. So how does it work? In this recent paper, they found that two different kinds of inhibitory neurons, through the link with the excitatory neurons, differently drive slow oscillation and fast oscillation. So together, that balance can produce or maintaining the normal temporal structure of local network connectivity. So this is very basic mechanism. Let's jump to the human level directly. We were talking about scanning brain, infants' brains. So this is infants from Yong He's group. Through collaboration with pediatric hospitals, this is infants, preterm infants. So they're about 31 weeks to 42 weeks post-menstrual. So this is like normally they should still in the belly of the mother. So they scan their brain. And look at this. So this is every 2 weeks, from 30 to 40 weeks. And the color shows the functional connectivity strength of the regions. So you see this drastic emergence of the functional connectivity hubs in the primary sensory motor systems. It's trying hard to develop all of those connections with the rest of the brain. And it's doing so by increasing specifically the short range and middle length functional connectivity. So we talk about critical period. So these are the critical period of the system to build. So we had started this very ambitious national consortium of children's studies. And it's part of the big China Brain Project that Fang was talking about. So it's also part of the ambition of that big China Brain Project, and BNU is playing a very leading role in it. So right now, there are 22 institutes in China participating already. We have constructed platforms for various data banks for Chinese children brain and behavior, and also genetic data, developing protocols for longitudinal follow-ups, aiming to understand these various ambitious scientific issues like biomarkers, sensitive periods, the effects of nature and nurture. I know it's big, but this is a big project. So the platforms that are ready. So we already constructed, recently published, Brain Atlas for Chinese children specifically. So this is based on the imaging data from 328 Chinese children, ranging from 6 to 12-year-olds, and to build a brain template that you can use as a reference point. It includes an age-specific map and also sex-specific map. It's important to do this because when we contrast this Chinese template with the NIH pediatric template, you see interesting differences with the frontal, especially frontal and parietal lobe. And also, when you analyze Chinese children's brain data, if you normalize to this atlas, you get better sensitivity and predictability compared to when you normalize to the NIH template. And then finally, so the platforms for the imaging analysis tools, for instance, we developed various kinds of, I say we, like He Yong's group and the Gong Gaolang's group, they developed analysis tools for looking at complex brain network data, for visualizing brain networks that's already used by 800 labs around the whole world, and not only applicable for human data, different kind of MEG, EG human data, but also mouse and monkey data. Okay, I hope you got the sense of the thing we did. When we are not in the lab working, we try to play with other research institutes. So this is one of the retreats we hosted in our Zhuhai campus. It's a very nice southern town. And next time if we host it there again, please come, because it's really pleasant. And I will end with an extremely short video from 8 years ago. So that was our president, Qi Zhong, speaking in the background. Thank you for Pat Laura. And the winter is leaving, the spring is coming. We have a bright future. That's very applicable for today. And I want to thank you especially to Pat, to Laura, to Hugo, the McGovern Foundation, the big McGovern Institute community involved. Thank you.
Thank you so much for that fascinating overview. Do we have any questions from the audience?
So the instrument you refer to is the piano. If you were to substitute that with a violin or flute or guitar, would that yield the same results?
I know exactly how to answer that question, Bob. That's his work.
Probably. That is a very unscientific
A very scientific answer. Okay, I actually asked the first author who always know the details of the work. She says, I was thinking, how does it work? Like learning to read really tells you about the auditory input as well. Why does piano works better? She says it's really the precision of the auditory cues. So I guess if you play piano is very good at directing that. With the violin, I guess you have to teach them really well to do this fine precision of the auditory, the pitch judgment. So pitch precision, I think is the key.
Okay, it's the piano.
Do a follow-up study, compare different instruments.
Any other questions?
So what are the key implications for drastically transforming the Asian educational system that's coming out of your research? Because I have been educated in Asia and in Europe and the United States, and I work globally with many young people that are products of those educational systems. And I'm wondering what the implications of your research are to maximize, if sensory is truly the key, and we have 5 senses, so we emphasize a few of the senses, especially in Asian memorization-based, test-based learning. Are there drastic transformations coming in the age of AI?
That's a very great question. I'm hearing different kinds of education styles, and also the AI. Did you say AI, artificial intelligence? Okay, a whole new era of AI. So there's culture difference, education styles, and AI in different senses, and the memorizing. So what's my take on this? I have to be very careful in answering grand questions like this, and I think we have some very indirect implications, like my work showing that senses affects where you store the knowledge, but it's not necessary in acquiring the knowledge. You're going to store it in the brain somewhere. You can acquire it. I think Rebecca's work with blind also showed that, right? But I think one thing that's more direct related to education is Lu Chunming's work, where they were recording from the teacher and the student and showing how the predictability of the teacher on the state of the students really matters. I think that's a big thing. In the classroom, like Beijing, you have like 70 kids in a classroom. You can't do that. Whereas here, you have more resources to do that. So I think, it's less than teaching style, but the availability of the resources allowing you to pay more attention to the individual. Or going back to Rebecca's work, like you have to be directing to the recipient. So I think these are the direct relationship. As for AI, yeah, I'm not comfortable like answering great questions without thinking too much. I don't see the direct connection there yet. Okay.
Any other questions?
I wanted to hear more about that amazing longitudinal sample. So how old are the children, and how many are there, and what is going to be studied about them? It seems like an incredible opportunity.
Yes, you should be part of it. Fortunately, I'm not part of it yet, so I don't know the inside information. And I'm trying to see if we have in the audience. Xinchang Zheng, yes, we have a PI here who is part of the program.
So I briefly mention this project because I was involved in this project since almost 3 years ago. As now the planning is, we already in the Beijing and also another city in the southwest of China. We have 3 sites. Now we have almost a complete second time point of about 600 from 600 children from 6 to 12, and this will expanded to over 22 sites across the whole China, and we're going to reach planning in next 5 years. And the first sample could be reached by 20,000. They're planning now for. And also depends on a Chinese brain project. how much like resources they were going
much
to
money
put
they
on.
get.
And including also Yanchao already mentioned that like we have a behavior and gene and also have the brain. So the 20,000 I means like including gene and the brain. But the behaviorally psychological measurements will be much larger. Like the president of our university already accomplished more than like it's about like 100. I think it's how to say it's like 100,000 for psychological measurements with behavior, but without the brain. For brain like data, we're going to reach like the current plans like 20,000.
But each thing I remember we're discussing whether it's realistic to go under 6, and we mentioned your work. So I think they're discussing it. Like very.
Yeah, this is I don't know much detail about this, but for the Chinese like children brain project will be collaborated closely with ABCD.
Okay, so we'll proceed to lunch in the poster session, and I'll see you all back here at 2 p.m. And I'd like to thank now all of the speakers this morning and the audience for your questions and attention.